My talk on the epigenomics of myeloma was primarily focused on describing several new assay technologies that are available in order to profile primary myeloma samples and what their epigenomes look like, particularly assays for DNA methylation and chromatin accessibility. Work from my group has recently described assays for chromatin accessibility, utilization in multiple myeloma to really understand how transcription factors, the primary factors that read the genome into the epigenome, how they are engaged with myeloma chromatin and how that is different from normal plasma cells. One of the major advances in myeloma therapy and understanding myeloma therapy over the past 5 years has really been, one, the identification of transcription factors as really primary targets of existing myeloma therapy. Dexamethasone is being targeting the glucocorticoid receptor, which has long been known, but especially the IMiD drugs, which have now been identified as targeting essential transcription factors in multiple myeloma. And so one goal that we have in profiling the myeloma epigenome is to try and understand, are there other transcription factors that may be essential for maintaining the myeloma epigenome that may also be good drug targets. The focus of epigenomic research in multiple myeloma could span lots of different types of avenues. I think a primary focus needs to be in understanding the heterogeneity of myeloma at the epigenomic level. We've known for a long time that there is quite a bit of genetic heterogeneity in myeloma. This also seems to be the case at the epigenetic level. And are those heterogeneous factors, do they correlate with the genomes of myeloma? Or are there different subgroups defined at the epigenomic level that may be prognostic for different treatment types or at the baseline level, essentially? I think, as with cancer and with normal cells, the action in terms of cellular biochemistry is really at the epigenomic level, and we need to advance epigenomic assays in order to define new biomarkers, define new targets for both epigenetic therapy and other types of therapy. And it's really a nascent and burgeoning field that deserves a lot of attention.